on the chromosomes. We're pulling out to a view which is as if you would look down a normal microscope. This is a living cell. It's under time lapse, so that's how you can see it all moving. The nuclear envelope breaks down, the chromosomes are released, and they go through this very striking sort of movement, which just seems to be all focused on, I've added these little red dots, they seem to be tugged and pulled to this equator position. And when the cell feels everything is ready to go, the chromosomes are ripped apart. One set of DNA goes to one side, the other set of DNA goes to the other, identical copies of DNA, and the cell then splits down the middle, creating two living cells, two daughter cells. So we're going to rewind and just focus on the DNA, the chromosome, and look at its structure. So again, here is that equator moment. It's the chromosomes are wiggling around, and I'm just going to isolate just one chromosome out of it, and we're going to have a look at its ultrastructure and how it's made. So here's one chromosome. Those things that are sticking out either side that look like whiskers are part of the cell's scaffolding. They're called microtubules, but they're for orienting the cell and for locating it, that center origin point. The sausages are, it's one chromosome. It's got two pieces of DNA. One's wrapped up in one sausage, the other one's the other strand of DNA. The red region is what we're going to focus on. It's a gigantic, huge protein complex which is made up of 100 to 200 proteins, and it is the interface between the chromosome and these long fibers, these microtubules. We've known about this red structure, I've labeled it red, for more than 100 years, and we have a lot of information about it. It is obviously central to the movement. We have no clue, really, as to how it's actually achieving the movement. But it's also a broadcasting system. It tells the cell that it's not ready. It's also a tension sensing system. It's able to tell through tension whether it is ready to undergo the separating of the chromosome. And it also is able to, it grows these microtubules, or it hangs on to the growing and shrinking microtubules. They're growing only in this particular image. So it's able to transiently couple onto the microtubules. It's turning green because it's sensed that the tension is just right. All the proteins get ready, and you'll see there's one little last bit that's still remaining red, that stop signal, and that is the signal broadcasting system. And it's literally walked away down the microtubules and away from the kinetochore. I mean, it's that mechanical. And so with a little bit of eye candy, molecular eye candy, these little guys are kinesins, are like little courier molecules carrying those little packages on their back. And here are the dynein coming from that kinetochore carrying that red broadcasting system. And they've got their long legs so they can step around obstacles where the other guys, they could. So the kinesin can only walk one way, dynein can only walk the other. So purpose of my talk is to really give you guys some feel for what molecular biology is actually like, and perhaps to woo some of you nanotechnologists and physicists over to the hot tub of biology. Because I have to say, the water is warm and it's bubbling, so come on over. Thank you.